Now we'll see the interval graph. If we have a set of n intervals on the line segment, then the interval graph is defined as the graph that correspond to the n vertices that are equivalent to the n intervals. And there are also the edges between the intervals, or we can say the edges between the vertices v and w. And the edges is only present when the v and w vertices they were overlap with each other. So these internal graphs, they are very easy to recognize because they have a very characteristic structures. For example, here we can see there are two types of graphs present. One is A, and A is an interval graph because it is overlapping with each other. In this graph we can see that there are 5 vertices A, B, C, D and E. If we consider the vertex A, we can see it is connected with B and C. If we consider B, it is connected with A, C and D and so on. And we can see very easily that these are overlapping with each other. For example, if we consider A again in this representation, we can see A is overlapping with B and it is also overlapping with C. So if we consider the gene B, and we know that it is overlapping with A, C and D, and we can see in this it is overlapping with A, overlapping with C and overlapping with D and so on. So when the vertices they overlap with each other, then the graph is called the interval graph. But we can see in the other graph that is present as a B, and we can see it is just like a house form, just like a house, and it is not the interval graph because there is no overlapping just like here in this graph. So we can say that the Benzer's problem was equivalent to deciding whether the graph obtained from his bacteriophage experiment represented a interval graph, and if he had the experiment resulted in a graph like this one, then the genes could not have been organized as linear structures. So as it turned out, the graph was indeed an interval graph like this one. So we can say it's indicate that genes are composed of linearly organized functional units.